So, hello everybody. I'm glad to present you our team supervised by Dmitro Fishman. My name is Natalia, here is Solomia and Sishko, and also one amazing volunteer, Oля, helped us during this project. So the project lies in the domain of bioinformatics, and actually it's an open problem. The goal of the project is to determine the rate of cancerous cells with respect to normal ones in breast tissue. And actually a little bit about the work itself. Most of the lecturers told you that 80% of time takes data preprocessing and only 20% of time takes data analysis. So guys, believe them. About the input data, we had 694 images that weighted 8 gigabytes. It was actually quite a lot for us, and we had to clean all this data, like to remove the white borders, to remove, oh, by the way, we had a CSV file where the doctors wrote their predictions about how the certain person is likely to have cancer or not. And actually we had to remove the data which did not correspond like one to the other in the file and in the data folder. Also we had to do the image scaling, like to compress all our data and to make it grayscale. As the output we obtained 528 images that weighted 17.5 megabytes, so that we could map it into a vector, like each image, and make a matrix out of it, so that PCA and different other data analysis tools are applicable to it.
So at this point of time, we transferred from the team of data preprocessors to the team of data analysts. And the first problems we faced with was the very imprecise estimates of the doctors for the new number of cancerous cells in the breast tissues. Here you can see the huge overlapping of all classes. Here's the very big class from 30% to 100%. It overlaps with this class and this class. So our solution was to split all these estimates into 5 classes first, which doesn't overlap between each other, and then to the 2 classes which also doesn't overlap with each other. In order to reduce dimensions of our data, we applied the PCA analysis to our data matrix, and as you can see, we also didn't get a big distinction between all classes. Then we split our data into training and testing set, and apply a current package to train our model. We used the 5 most popular algorithms from current package, and here you can see the results on the testing set for our 5 classes and for 2 classes. And we put the emphasis on the splitting into 2 classes, which give the accuracy 63.5% for each GBoost linear algorithm. So the next slide shows you, sorry, it's like Super Mario style. You can say, okay, haven't you visited the lecture of data visualizing, but it's still quite informative. It helps us to visualize our accuracy of our model, and here the black line is the most accurate XGBoost linear model, which gives our accuracy 63.5%. And where the random one is 1/2.
So, ladies and gentlemen, in conclusion, I'd like to say next speech. So, as a result, we improved our skills in R, and also we learned how to make accurate right data preprocessing, which means to put all of this ugly data into readable format. While preprocessing, we faced a lot of obstacles, like too big variables and also unclear images with drops of solutions which biologists use to determine cancer cells, and also really unclear classes distribution made by doctors, really lazy doctors, as you saw. Also it was really interesting just to explore all of these data analysis features in R, especially if you're new to this language. But we braced up and overcame all of these challenges. And also it would be interesting maybe just to use different approaches for solving our problem, and it's like deep learning frameworks, and then probably we would receive other results.